my talk is about the nanopore electrochemistry for a single molecule. Maybe I heard your lectures today. That's great. And my research is about chemistry, but first of all, I want to share some experience for an editor for the chemical science. And most of the submission from the electrochemistry and the science about electrochemistry that come to me. So I'm the only associate editor for chemical science to handle the analytical area manuscript. So please support this journal and submission to us. So that's my research in Nanjing University. It's about interface, and we're doing the single entity electrochemistry and the nanopore electrochemistry, which I want to talk about today. And also we're doing some instrumentations and big data analysis. So that's all my group in Nanjing. If condition allowed, and welcome you to visit Nanjing anytime you wish. And for my research, I started my PhD in Nanjing University 20 years ago with Professor Hongyan Chen. He's an electrochemist in China. So that time we were working on the metal electrode, which I consider is a one-dimensional surface, which means it only can determine the concentration of an analyte. So when I go about outside of China, and in Canada, I studied the model layer on the surface, which is like the grass on the ground. So I think we modified the DNA on the surface. So I consider this one is a two-dimensional surface because this the metal is on the bottom and the molecule on side on the electrode surface. So we sensing the DNA strength on the surface. So this is a chemical modified interface. And when I back to China, I studied a surface which is coating the lipid on the surface. The lipid we know is a soft material, so molecule can move around in the lipid layer, inside the lipid layer. So we can coating by lipid layer on the surface. So the intact sites such as coenzyme can move around in the lipid. This is biomimic surface. We call this confined biological environment. So this is three-dimensional. It's a space on the surface. And now we are working on this nanopore study. Nanopore, which is protein inside the membrane. So the target molecule can inside the channel, can pass through the channel. So there will be time series information including. So we call this four-dimensional, including a time series interactions inside the channel. So we will talk about it soon after. So running this one, we say this is a confinement electrochemistry. So on the, like I talked about before, the micro scale, which is electrode or particle interface, even capillary or pore. And the energy looks, there will be electrical energy and electromagnetic energy or fluidic field, which is geometrical space plus time series energy. This we call this confinement effects. For the nanoscale, there will be the energy will come to the chemical bonds, chemical group, and the weak interactions such as hydrogen bonding interaction, the network. We can control that biochemically. So let me talk about the electrochemical confinement here. For the traditional big electrode, we can get the CV curve to study the molecule. If this is the size of the electrode down to smaller, then we can compare with the single particle size, so we can determine the single particles. So for less measurement, we need a higher current measurement, lower current measurement, and higher the higher bandwidth. So we do some small simple calculation. Under this condition, we need to measure several electron or several charges changes. So I think Christine long time ago showed some the information for this high bandwidth is very important for electrochemical measurements. And here we just do some calculations and to see the filter is very important to measure the current. So we developed some ultra sensitive instrumentation in our group. That's the chemist doing the electronics in our group for almost 10 years. So we can measure the tanning current with higher bandwidth. So with this instrumentation, compare the commercial one, we can get more information. In the commercial one, we only can get a single peak, but for our measurement, we can get multiple peaks for the same electrochemical system. So we confine we were on the surface, we can confine the collection measurement, use the server and coating electrodes. And also we can modify the monolayer, the TiO2 film on the surface. We can look at even the faster electron transfer on the surface. So all of this one that depend on the electrical instrumentations, and even we can review the potential on the solution. So in a traditional electrochemistry, the potential can change only by potential state and pH or and the solid state, solid sort of the electrolyte, but not affected by the tag molecule. Here we just show example when a tag molecule is big enough, so there will be the potential will be infected by this tag molecule, tag electrolyte tags here. So we doing some the potential distribution on the electrical surface. That's only by this sensitive instrumentation we can see this informations. So then back to the nanopore. So consider nanopore on the surface, on the bilayer. This is a bigger resistors. So the drop potential like 100 millivolts, there will be 99 millivolts will be dropped down on the pore. So the huge electrical field were on this pore side. So consider the traditional electrode surface. So we drop the potential in the solution, and the electrochemical reaction happen on the surface of the electrode. So level can get the server CV voltammetry, but when we pour on we drop the potential on the pore. So this area is bigger resistors. So all the potential drop on this pore, like I talked before. So and we can this will be enhanced the resistors until the pore tip. So we will recording the current with the time. Also, this means the current with the voltage, same as the CV voltammetry. So this we think the nanopore is the electric chemistry as well, and the pore is the electrode surface as well. So then back to the traditional sensor interface. On the sensor interface, we modify the molecule on the surface, but the tag interaction, this one, or what's the size between this one is hard to control. So can we make only one molecule on the surface? So think about this question when we think this planned surface will be turned over, become a cylinder, then it's a nanopore. So this nanopore is one molecular protein. So it's a one molecular interface. So that's the biological nanopore. So this biological nanopore is formed by amino acids. So we can see this is only 1 nanometer the size of this opening. So this when the tag inside the molecule, inside this channel, they will pass through this channel, they will interacting with each of the amino acids here. So this we call this one is a single biomolecular interface. This is the geometrical of the pore. It looks like our nose or ears. All those organs that's for get the noise, get the sensing the noise, sensing the smell. So for the biological nanopore, all those amino acids can be changed by the genetic engineering technique. So on the top of the pore, this will be sensitively region. So the target there will be selectively target molecule, and for this the channel, we think is all this one is sensing area. So this sensing area will be sensing the molecule by each of the amino acids. So we can play with charged, non-charged, and hydrophobic and hydrophilic amino acids to interaction with the target molecule. So with this idea, we studied the peptide structure and also the aggregation of some the interesting amyloid. And at this stage, we can challenge the amino acid sequence. So all the 20 amino acids, when they will be interaction with this amino acid, we designed the sensing area, so we can discriminate those amino acids by sequence. So and for some same molecular weight amino acids, this is harder to be determined by the volume, but we can determine that interactions. So this we can use the standard deviation current blockage. So we can look at this small change in the current, so we can determine those kind of peptides. Even the only one amino acid changes, we can determine that. So like amyloid, this only A and to G difference, we can determine that inside of the peptides. And back to this nanopore, when the molecule passes through this nanopore, they will interaction with ionic current. So all those water interact, all those water inside here as well. So they will can form the network of the hydrogen bond. So we think is interaction network will be inside this channel. So all those data information can be determined by current. So those current looks like a noise, but this information hiding this noise. So we can use the software to review these interactions. Here we use the Herbert-Huang transform to extract the frequency information from these nonlinear, non-stationary signals. So we can track the information from those current and get the different understanding for this network. We can play with the different mutated channel to look at the different peptides here. So as the conclusion, we can get the frequency fingerprint to describe the dynamic interactions inside this nanopore. So this interactions looks like the piano, and we can play with the keyboards. Then we can get the music of the molecule. So for the higher resolution electrical instrumentations, we developed for 10 years. So we can push down the hard bandwidth down to no higher than 100 kilogram. So for the we also can we can get a higher aperture for the high bandwidth instrumentations. We can compare the instrumentations. The error is less than 0.1% for different pores. So also we have the data recording and the data process software to put online to use for you. And also for this technique, if we only on my level is not this like FE race car. So we use the Volkswagen. Let's everybody can drive that. So I use this technique to be used in the undergraduate student class course, the experimental course. So undergraduate students in the second year, they can do this one in one afternoon. So this technique can be repeated every chemical laboratory. So come to the end, I want to get a shorter conclusion here. So we studied the single entity electrochemistry, and while it's nanopore electrode, all the pore surface, it's the electrode. That's my understanding at this moment. Also, when this pour down to the single molecule, like biological protein, so this channel, we think it's also is the electrode surface. It's called a single molecule interface. Here I organized the meeting called Nanopore Electrochemistry from 2020 because the COVID-19 happened. So we can only online to see all the friends and scientists here. So even Nature Chemistry reported these events. And these events continually for 3 years already. So all the postdoc students and the research in this area, they can join these events. So for the Friday discussion, also I had a series on the meeting on the electric chemistry, and for the nano side, 2016 in New York, we Justin proposed the single entity electrochemistry, and 2018 in Bath, we talk about the electrochemistry at a nano interface, and the 21st in China. But this online meeting, unfortunately, we talk about the next generation of nano electrochemistry, and for 2024, we also proposed this meeting in China. Hopefully, this is the new horizon in nano electrochemistry. So welcome all the friends and the scientists to visit us in Nanjing. So thank you very much. Sorry for this delay and for the technical issue.